Okay, I think we're good to go. Hi, everyone. Welcome to GemPharmatech webinar of the month. My name is Abby Zhu. I'm a client manager at GemPharmatech, and I will also be your host today. It is my great pleasure to introduce my colleague, Dr. Jianming Xu, to share his experience and knowledge in genetically engineered mouse models for preclinical evaluations. GemPharmatech is specialized in engineering mouse models using cutting-edge gene editing technologies. As a brief intro, Dr. Xu is a research scientist at the GemPharmatech R&D department. He joined GemPharmatech in January 2021. He obtained his PhD degree in biochemistry from the University of Munster. He has extensive experience in the regulation of gene expression in the mouse embryonic stem cell and small molecules targeted anti-tumor therapeutics. Prior to joining GemPharmatech, he worked at German Cancer Research Center and the University of Zurich. He and the R&D team of GemPharmatech strive to provide innovative mouse models to our customers in order to meet their needs of preclinical drug screening using our scientifically comprehensive and reliable data and professional dedication. Now, everyone, please welcome Dr. Jianming Xu.
Hi, thanks, Abby, for the nice introduction. This is Ming here, so I'm glad today to discuss with you about our human immune system reconstituted NCG models for immunotherapy. So why do we need humanized mouse models? So first of all, the laboratory mice are very useful tools for preclinical studies because they are very small and they grow quite fast. So it's very cost and time and cost effective to use the mice as a preclinical model. However, for certain applications, like certain therapeutics, which is based on antibodies, they only recognize the human proteins. So in this case, the mouse cannot fully recapitulate the human situation. That's why we need to use humanized mouse model for preclinical research. Just to give you an example, cancer immunotherapy has received increasing attentions in the past decades. So here are a few lists of therapeutic antibodies which either have been approved or in clinical trials for cancer treatment. And most of these antibodies, they recognize human epitopes. That's why to more precisely reflect the human situation, we need the humanized mouse model for those antibody-based therapeutics. So speaking about humanized mouse models, there are two different types of humanized models. One is the immune reconstituted models. So in this case, the entire immune system will be human, and the human immune system will be engrafted into immune compromised mice. And this is the topic of today's webinar. However, there's another kind of humanized models which use the mouse cell line and mouse immune system. However, the therapeutic target is humanized. So the focus of today's webinar will be on the first group. So the foundation of GPT's immune reconstituted model is based on our proprietary immune deficient mouse model, which is NCG. We can do two different types of immune reconstitution. So either with human PBMC or with human HSC. So there are pros and cons for these two types of immune reconstitution. and I will elaborate more in the following slides. And also in the second part of this webinar, I will introduce to you our newly developed next generation NCG models. So they have great applications in different area. So first, I would like to introduce to you our main player for today's webinar. This is NCG, and this is our immunodeficient mouse model, which supports the whole Immuno-Oncology platform. So the picture here shows you the evolution of immunodeficient mouse models. So the wild type mouse has an intact immune system. Then we have the nude mice, which is also known as the acymic mouse. So there's a gene called FOXN1, which is mutated in these mice. Therefore, the development of the thymus is compromised. And also those mice, they are hairless. Then it is also, it has been discovered that certain mutations would lead to a compromised T cells and B cells development. So that's the not SCID mice. However, in those mice, we still have the remaining NK cells and myeloid cells. So for a complete immune deficient mouse model, we and others have developed the next generation immunodeficient mouse model, and these are the NCG and from GPT and NOG and NSG and several others from other distributors. And those mice are ideal model for human cells engraftment. So the NCG is developed by GPT, and it is distributed by Charles River for European and North American markets. So what is NCG? So there are two genetic modifications on the NOD background. So one is the PRKDC SCID mutation. So this is a mutation initially discovered in the BABC background. And this mutation would lead to a deficiency of T cells and B cells. And it is known that different backgrounds shows different severity of the mutation. And in NOD background, this mutation works pretty well. And in addition to it, we also created the IL-2 gamma receptor by our CRISPR-Cas9 technology. So this would lead to depletion of receptors which are using the IL-2 gamma receptor, and this would lead to the depletion of NK cells. So now let's talk about immune reconstitution. So as I mentioned previously, there are in general two different ways to how we can do the immune reconstitution. So one would be with the human PBMC. So the human PBMC stands for peripheral blood mononuclear cells. So these are very specialized immune blood cells with round nuclei. And they were developed in the bone marrow and located in the peripheral blood where they can be isolated. So our standard procedure for human PBMC reconstitution is we take PBMCs from donor, then we inject those PBMC into our NCG mice. So either separately or together with cancer cells. So it depends on which cancer cells we are using here. Then we have a human PBMC reconstituted NCG mice bearing the tumor, and this would allow us to do preclinical drug efficacy test. So the key advantage of human PBMC reconstituted NCG mice are as following. So first of all, the reconstitution is very fast. Usually the reconstitution can be done within 1 to 2 weeks. And mostly T cells will be reconstituted. And the lifespan of human PBMC reconstituted NCG mice are around 4 to 7 weeks due to GvHD reactions. And we can establish a donor bank. Each donor can support roughly 200 to 300 mice. So here are data showing how good is our PBMC NCG reconstitution. So as you can see, within the first 2 weeks, we can reconstitute more than 20% of the leukocytes as marked by human CD45 cell surface marker. And most of those cells are actually CD3 plus cells, so which means most of them are actually T cells. And this can be done within 2 weeks. So here is a case study we have performed at GPT. So we used the human PBMC reconstituted NCG mice, and they are either inoculated with a breast cancer cell line or a colorectal cancer cell line. And we evaluated drugs which are targeting the human PD-L1 in these 2 different models. And we can observe a tumor regression after 4 weeks treatment. So here is another example. In this case, we used a T cell engagers, which is a bispecific antibody. So it recognizes both the tumor cells as well as the T cells. So it engages the T cells to target the tumor cells. And this is a collaboration study between GPT and our partner. And this has been published last year. So the working flow are showing here. So in this case, we actually co-injected the human PBMC together with the cancer cell line into our NCG mice. Then 7 days after we started the antibody re-injection, and then we have 3 weeks observation time. And the figure shows here is the tumor volume developed on time. And you can see that with different isoforms or different modifications of this bispecific antibody, we achieved a very good tumor regression or tumor inhibition. And here are individual data. So as I just mentioned, so one problem with human PBMC reconstitution is the short observation time window. And this is mainly due to the GvHD reaction. So this is what we can show here. On the left side is the mouse body weight, and on the right-hand side is the survival rate. So as we can see here, so roughly 6 weeks after the reconstitution, we started to see mice dying, and to different extent. And we also performed the pathological analysis of the mice, and we can see that human immune cells infiltrated into different tissue of the mice. So here are some pictures we took from those mice which suffered from a GvHD reaction. But not everything is bad, because the human PBMC reconstituted mice shows an early GvHD reaction. So those mice actually can be used to evaluate anti-GvHD therapy as we performed, and we show the data here. So with the treatment of anti-GvHD treatment, we can significantly improve the mouse survival rate. So now I'm going to talk about the second type of human immune system reconstitution. So this is done instead of using the human PBMC, here we are using human HSC. So what is HSC? So these are hematopoietic stem cells. So you can consider this as the progenitor of all blood or immune cells. So these cells are multipotent and they can differentiate into various cell types. So those cells are developed in bone marrow, and they are found actually also in the peripheral blood and also the umbilical cord blood. And this is actually where we got those cells, and they are very often used in translational therapy. So the key advantage of human HSC reconstitution is that in comparison to the human PBMC reconstitution, they have a significantly longer observation time window. So usually, these mice, they last longer than 16 weeks. So this would allow us to do a longer treatment. And also the reconstitution time frame is much longer than the human PBMC. So it generally takes from 8 to 10 weeks to have a reconstituted mice to be used for preclinical studies. And the procedure looks pretty much similar like the human PBMC reconstitution. So we first inject the human the human HSC, which is marked by CD34 plus cell surface marker. So those cells are injected into the NCG mice. But prior to injection, we normally do either irradiation or with chemicals such as busulfine to deplete the residue mouse HSC, so that the engraftment of the human HSC would be much better. So then after around 8 to 10 weeks, when the human immune system is reconstituted, the cancer cells can be inoculated. So either subcutaneously or also topically into the reconstituted mice. Then we can have a human immune system reconstituted mice bearing actually a human tumor. So as you can see here, the survival curve of the human HSC reconstituted mice, so they survive much longer than human PBMC. And here are our experiments. We show how good is the human HSC reconstitution. So typically we have a cutoff at around 25% of human CD45 plus cells. And we can see that both actually T cells and then together with some B cells can be reconstituted in this model. But the B cells here are actually not functional. So it also mainly reconstitute the T cells. So here is another case study we did using our human HSC reconstituted NCG mice. So in this case, we also used a T cell engager. In this case, this is a therapeutic antibody which targets both the CD19 and the CD3. And in this case, we inoculated a human lymphoma cell line called Raji, and we performed the antibody treatment. So as we can see, with two different therapeutic antibodies, we achieved quite significant tumor regression after 4 weeks of antibody treatment. So here is a brief summary for what I have showed you up to now. So we have developed the severe immunodeficient mice called NCG, and with those mice, we can do human immune system reconstitution. So either with the human PBMC or with human HSC. And the key difference are the different reconstitution time. So for human PBMC, the reconstitution is pretty fast. So it can be done within 2 weeks. However, for human HSC, it generally takes around 8 to 10 weeks until the mice are ready for tumor inoculation. And then another key important difference is the survival time or the lifespan, so to speak. So with human PBMC reconstituted mice, so due to an early GvHD reaction, so these mice usually they don't survive longer than 7 or 8 weeks. So there's a short observation time window. And for the human HSC, these mice survive much longer, usually up to 18 weeks or even longer. So this is very suitable for a long time treatment. And in addition to the NCG mice, we also developed several strains based on the NCG mice, and we call them the next generation NCGs, because there are some disadvantage or shortcomings with the first generation of NCG. So mainly they can only reconstitute T cells. So for customers or clients who are interested in NK cells function, so we also developed the NCG human IL-15 mice. So these mice would support the human NK cells reconstitution. And also we have the NCG SGM3 mice, and these mice support the myeloid cells reconstitution. Then we also have the NCG with MHC class 1 and class 2 double KO mice. So this would significantly improve the human PBMC engraftment, because these mice would suffer much less from the GvHD. And the last but not least, we also developed the NCG-X mice. So those mice are different from the NCG in a way that no irradiation or chemical treatment is required. So they are ready for human immune system reconstitution. So first, I would like to briefly introduce our NCG human IL-15 mice. So we know that the human cytokine IL-15 plays a pivotal role in the development and differentiation of NK cells from common lymphoid progenitor cells. And also the NK cells recently also becomes a hot topic for cancer immuno-oncology. So there are a lot of therapeutic antibodies are actually targeting NK cell receptors. Therefore, there is a demand of in the society, so how one can reconstitute NK cells with immunodeficient mice. Therefore, we developed the NCG human IL-15 mice. So in these mice, we replaced the mouse IL-15 with the human version. And here are some validation data. So on the left side is the mRNA level of the IL-15 expression between NCG and NCG human IL-15. So as you can see, the tissue specificity and expression level are pretty similar actually between NCG and NCG human IL-15. So on the right hand side, we measured the human IL-15 protein level by ELISA. So you can see that both the NCG human IL-15 heterozygous as well as the NCG human IL-15 homozygous mice showed a quite decent expression of the human IL-15 protein level. So we can use those mice to reconstitute the human immune system with the focus on human NK cells. So we have different strategies how we can do this. So we can either inject the human PBMC or human HSC as we did for the NCG mice. So in particular, in this case, we can also pre-purify the NK cells from the blood. Then we can inject the purified NK cells to those mice. So here is a comparison between the human PBMC versus NK cells reconstitution using our NCG human IL-15 mice. So as you can see from here, the human PBMC can reconstitute mainly the T cells, and the level of the NK cells is actually pretty low. But with purified NK cells, with purified NK cells injected into the NCG human IL-15 mice, we can maintain a very decent percentage of the NK cells. So this would support, this means our NCG human IL-15 mice can support and maintain the NK cells in vivo. And here is the comparison of the NCG versus NCG IL-15 mice in their survival rate, so and body weight. So in addition to the PBMC and the purified NK cells, we can also do immune system reconstitution with human HSC. So here are our validation data we performed in our internal quality control. So we could show here that the NCG mice usually doesn't support the NK cell reconstitution with human HSC. However, with our NCG human IL-15 mice, the reconstitution rate of human NK cells are way much higher than the NCG mice. And also we did a in-depth analysis of the NK cells reconstituted with our NCG human IL-15 mice, and we can show that with different cell surface marker, we can show that different subgroup of the NK cells indeed can be reconstituted. So this is another collaboration study between GPT and our partner. So in this case, we did a preclinical efficacy study using our human HSC reconstituted NCG IL-15 mice inoculated with 2 different types of tumors, and we tested different therapeutic antibodies. So as we can see here, the tumor regression, we obtained a quite decent tumor regression after a long time drug treatment. And the NK cells are very well known for its function in antibody-dependent cytotoxicity. So with our NCG human IL-15 mice, we also performed both in vitro and in vivo analysis. So in vitro, we purified the NK cells from the reconstituted mice, and we performed an in vitro ADCC killing experiment. So with different ratios between the NK cells and the tumor cells, we can observe a significant antibody-mediated cytotoxicity. And this correlates with an in vivo tumor regression with our HSC reconstituted NCG human IL-15 mice. And next, I'm going to show you our NCG-X mice. So as I told you before, the use of the NCG mice for human HSC reconstitution generally requires either irradiation or chemical treatment to deplete the mouse hematopoietic stem cells. And therefore, we introduced additional mutations on top of our NCG mice. So this is the c-KIT mutation. So this mutation would lead to a depletion of the mouse hematopoietic stem cells. So it's a point mutation from G to A, which leads to a valine to methionine mutation. And we also compared the reconstitution efficiency between irradiated NCG mice and NCG-X mice without any irradiation. So as we can see here, the general reconstitution rates were even higher with the NCG-X mice, and the T cells and the B cells are very much comparable between the NCG and NCG-X mice. However, it is known that the NCG mice doesn't support the erythroid reconstitution very well. However, with the NCG-X mice, we can significantly improve the erythropoiesis. This means we can have a large fraction of human erythroid cells reconstituted with our NCG-X mouse model. So next comes the NCG-SGM3 mice. So these mice, in these mice, we transgenically expressed additional 3 human cytokines. So this would support the myeloid cells differentiation. So as you can see from here, so we compared the NCG-SGM3 with NCG mice. So the general reconstitution of T cells are very comparable, with human CD45 cells slightly higher in NCG-SGM3 mice. However, the most striking difference comes from the myeloid cells. So namely the granulocytes, neutrophils, dendritic cells, and macrophages. So those cells can reconstitute very well in the NCG-SGM3 cells compared with the NCG mice. And this is a case study we performed with our collaborator. So in this case, we tested the combined treatment of anti-human PD-1 with a therapeutic agent provided by our partner. And we observed actually increased tumor killing effect with a combination of treatment using our NCG-SGM3 mice. And here are the endpoint analysis of tumor infiltrating lymphocytes. So we analyzed the TILs in the tumor. So last but not least, I would like to also to highlight our so another next generation NCG mice. So this is the NCG MHC class 1 and class 2 double KO mice. So as I showed you before, the human PBMC reconstitution with NCG mice suffers from the GvHD reaction, and the GvHD reaction is caused mainly via the mouse MHC class 1 and class 2 molecules. So in this mouse model, we knocked out both the MHC class 1 and class 2 molecules from the NCG mice, so that after human PBMC engraftment, the GvHD score is significantly lower compared to the NCG. And this leads to a increased lifespan or survival rate of those mice. So now let's come to the summary slide. So I have showed you that with our severe immunodeficient mice, NCG mice, we can do human immune system reconstitution, either using the human PBMC cells or with human HSC cells. However, with these two strategies, it can only reconstitute mainly the human T cells. In most cases, this can be enough, but for many different applications, such as therapeutics targeting the NK cells, or clients who are interested in myeloid cells reconstitution, or those who are interested in erythroids, we also developed our next generation NCG models. So this model can meet the need of different clients or customers in different immuno-oncology field. And we are not, we did not stop here. So in our pipeline, we also have several other strains under development. So soon we will have all of them being validated, and we can have a large variety of NCG-derived strains to meet the different needs of different customers. So that's all from today's webinar. Thank you for your attention, and I'm happy to take questions.